Recombinant DNA technology is a process in genetic engineering by which DNA can be cut and recombined. Scientists use this technology to develop vaccines, create protein-based medications, and create genetically modified organisms. To recombine DNA, it must first be cut. Degradative enzymes called restriction enzymes act like molecular scissors. They recognize specific DNA sequences and cut the nucleotide strands based on those sequences. Where the enzyme makes these cuts are called restriction sites. Some restriction enzymes, like EcoR1 and BAMH1, make cuts that leave jagged edges called sticky ends. Other restriction enzymes, like HEY3, make straight, clean cuts called blunt ends. Sticky ends are usually used to create recombinant DNA molecules. Because the restriction enzymes cut at specific known places, scientists can use them to cut a gene of interest, such as the gene that codes for the production of insulin from the rest of a DNA strand. Plasmids are rings of DNA in bacteria that can be vital tools in genetic engineering because they can be manipulated to carry a gene of interest into a cell that will produce the protein of interest. A plasmid is cut with the same restriction enzyme that is used to isolate the gene of interest. This cut creates an opening in the plasmid where the gene of interest can be inserted. Once the DNA and plasmid are cut, the gene of interest can be inserted into the plasmid. The gene of interest binds to the open plasmid through complementary base pairing. After insertion into the plasmid, a linking enzyme called DNA ligase acts as a kind of glue to ligate or join the DNA. The result is a recombinant plasmid. The plasmid can then be inserted into a cell, acting as a vector to carry the gene of interest into the cell. This is where the protein of interest will be made. This protein can be used to create a vaccine, medication, or genetically modified organism. As the cell containing the plasmid reproduces, the plasmid is copied, and so is the gene of interest, which increases the production of the protein of interest. Not all ligations are successful. Sometimes, the plasmid ligates without the gene. This is called self-ligation. Because the gene of interest binds through complementary base pairing, it can sometimes go into the plasmid backwards when it is inserted. This is called an inversion. If the plasmid undergoes self-ligation or an inversion, the gene of interest will not be expressed in the cell, and the protein it codes for will not be produced. Think of it like a circuit. If you connect a light bulb to a battery in the correct orientation, it lights up. But if you are missing a piece, like the battery, or the wires are connected incorrectly, it won't. Using recombinant DNA technology, genetic engineers can cut and recombine DNA. This manipulation allows for development of many things that impact our daily lives, including vaccines, protein-based medications like insulin, and genetically modified organisms, such as some of the foods we eat.